All right, and welcome to the webinar, everybody, today. We're going to be going over high content analysis and image cytometry as complementary approaches to advanced infectious disease research today. What we'd like to show you is a little bit about what PerkinElmer and Novo software have been collaborating on in terms of instruments and data analysis software. And we have some great presentations today that are coming from Gianluca and Krishna of the USAMRIID to show us how they're using these technologies, these instruments, and these data analysis platforms to do some very striking research. So today, Seungtaek Lee from PerkinElmer is going to be speaking a little bit about the PerkinElmer side of things and high content analysis. My name is Burke from Novo software. I'm going to be speaking to FCS Express Image Cytometry, which is going to be taking some of those results, image cytometry data from PerkinElmer devices to analyze that data in a flow cytometry style manner. And then again, Gianluca and Krishna from the USAMRIID are going to be presenting on what they've done with that data. So Seungtaek, if you'd like to start here, let me make you the presenter, and you can talk a little bit. Go right ahead.
Thank you, Sean. Hi, everyone. My name is Seungtaek Lee from PerkinElmer. I'm the high content screening informatics specialist for North America. Thanks for joining today. So I'd like to take a few minutes to go over the imaging technologies that PerkinElmer provides, and of course focus on the technologies that the USAMRIID speakers today, Dr. Pegoraro and Dr. Kota, are presenting on. So PerkinElmer provides a variety of high content and imaging tools, as you can see on the screen right now. So USAMRIID, they are currently using our Opera system. Our Opera system is a ultimate in high content imaging. The main benefits of the Opera are its high resolution from the water immersion objectives that we use, as well as the Yakugawa Zinco spinning disk that we implemented. And also we get the true benefit of high throughput due to the 4 CCD cameras that are running simultaneously to capture 4 channels at the same time. And along with the Opera, they use the acapella image analysis software. They also have the Operetta instrument, and this is a lower cost solution, and mainly I believe USAMRIID are using this for slower or lower throughput screens as well as assay development. And along with that comes our Harmony software to run your high content image analysis. And they also have our Velocity software. Our Velocity software focuses on analyzing 3D and 4D image sets. So in cases where they're taking high-resolution Z-stacks and they want to reconstruct the image in 3D, 4D, and measure any part of their images in 3D using voxels, they've been using Velocity for that. Okay, and we also provide 3D live cell imaging capabilities with the UltraView. And also, all of their images at USAMRIID are being managed by what we call Columbus, our image management and analysis system. So Columbus is able to manage, store, and analyze images from all different instruments. Obviously, our instruments, our Opera, Operetta, and UltraView, as well as all other high-content screening imagers and scanners, as well as microscope images. So once all those images are pulled into Columbus, you have one point of storage, and you can view and analyze all your images using your favorite browser. Okay. And now I would like to focus more on some of the application that our instruments can provide and our software can provide. Okay, so this is just a quick overview of all the different ready-made solutions or applications that we provide with our instruments as well as with our software, including Columbus. So this is just to give you an idea of some of the commonly used applications, but obviously we can create, or our customers can easily create their own ready-made solutions or analysis routines. And our customers and scientists can build analysis routines using what we call building blocks. So here are some of our building blocks that our customers can use to build their own customized image analysis routines. So here on the left side are some commonly used segmentation building blocks. So fine nuclei, cells, cytoplasm, spots, etc., as well as some calculation building blocks. And recently we launched an advanced version of Calculate Morphology, as well as Calculate Texture, which really helps in focusing on phenotypic analysis. And as we know that screening and drug discovery has been focusing more on phenotypic assays and phenotypic drug discoveries. So Dr. Pegoraro and Dr. Kota will be speaking about how they're using our morphology and our texture analysis tools to get more information out of their images. And to talk a little bit more about texture and morphology, this is just an example of how we're able to calculate texture. So here I'm showing you a screenshot of what we call SCR features, which stands for saddles, edges, and ridges. And we're able to measure these type of patterns, spot-like patterns, ridge-like patterns, saddle-like patterns, and etc., on individual cells. So these are intensity-independent patterns or textures that we can measure, and that gives you a really nice score that represents what's going on in the cell. And again, it's independent of intensity. And this is just an example of what are some of the masks or images that are generated from our SCR texture. So you can see an example of tubulin staining in the upper left in red, and all the other black and white images are filtered images or masks generated from our various texture analysis. And along with the texture masks, you also get a score that represents how ridge-like your staining were in your particular cell region. For example, cytoplasm, nucleus, membrane, etc. And again, Dr. Pegoraro and Dr. Kota will be using these techniques to quantify their phenotype. The second set of features that our scientists from USAMRIID will be showing you today, it's called star morphology. So these are new advanced morphology features that we added to our Columbus system, as well as our instruments, to measure some advanced morphological features, such as symmetry, threshold, compactness, axial, radial, and profile. I'm not going to go into detail into all of them, but maybe just a couple that Dr. Pegoraro will be using today. One is radial, and radial will characterize the intensity distribution in radial direction. So analyzing the fluorescence distribution in radial direction. So again, this is independent of absolute raw intensity. We're looking at the radial distribution of intensity. So this is one of the key features that Dr. Pegoraro will use today, or show today. Second is profile. It's another star morphology feature where we're looking at, again, fluorescence intensity distribution within a certain region, predefined region of a cell. So here we can focus on whether your intensity distribution is near the edge of the well or membrane, near the center of the cytoplasm, near the ring area of the nucleus, immediately inside of the nucleus, or at the center of the nucleus. So this is another star morphology feature where we can monitor your intensity distribution within any cell region. And it's very intuitive to apply these building blocks. So again, using our Columbus system, you can add modules in sequence. For example, here on the screen, you can add find nuclei to find all the nuclei within your image, find cytoplasm to determine the cytoplasm border, select population, perhaps to filter out the cells that are dying, and then calculate some statistics or morphologies or phenotype. For example, you can add a simple calculate morphology building block, calculate intensity building block, calculate texture building block. So we just need to add these building blocks in sequence, and it will add all the attributes pertaining to those building blocks. And in addition to Columbus, we provide a cappella, which I mentioned earlier. This gives our customers another platform where they can build very advanced and very focused algorithms. So this is a script-based language. So for customers who are familiar or are comfortable using a script-based language to build their own image analysis routine, they can do so using a cappella. Last but not least, obviously, we're trying to show how high content analysis and flow cytometry-like analysis complement each other. And Columbus has been the hub or a key tool in that complementation. For example, here, any HCS imager or microscope images can be imported into Columbus. And once you run the image analysis in Columbus and obtain all the numerical and cellular data, they can be exported to FCS Express. And then you can run your image cytometry or flow analysis on the FCS Express software. And that's what Dr. Pegoraro and Dr. Kota will be showing you today. So again, Columbus is acting as a hub for all your image data. So whether they're from HCS or whether from microscopy, you can analyze it in Columbus, obtain all the cellular data, and then send it to FCS Express for flow-like analysis. With that, I would like to take it over to Sean.
Great, thank you, Seungtaek. So I just wanted to pick up where Seungtaek left off, whereas the kind of exporting from Columbus to start working in FCS Express. So I wanted to start by telling you a little bit about FCS Express first. FCS Express has been around for many years, and it's been developed as a powerful flow cytometry data analysis and presentation software. And in the last 2 years, we've been very proud to release our image cytometry product. The main reason is because the main principles of flow cytometry and image cytometry overlap. Such things like taking multi-channel fluorescent data, plotting that data in scatter plots and histograms to look for statistically significant differences and changes. But what we can now do is take that image cytometry data that you're getting from any of the Columbus centralized places or instruments. You can take that data and leverage the most powerful analytical tools that we have in our flow cytometry package to analyze those multi-parametric imaging data sets. And what we can do is relate those multi-parametric data sets directly back to the original images, to wells in a plate, to single cells for confirmation of those results. So in the end, FCS Express image cytometry is going to allow you to spend a lot less time reviewing these really large, overwhelming data sets and analyzing kind of raw microscopic images by using these tools that have been proven to handle data sets with millions of points, lots of different phenotypic combinations, and you're going to have, again, access to the raw images when you need. What I first wanted to show you real quickly is the workflow of FCS Express. Seungtaek kind of touched on this. Again, what we do in FCS Express is just the data analysis side of things. So we're not doing any kind of image acquisition and image segmentation because other people are very, very good at that. Just like PerkinElmer and Columbus and Acapella and Building Blocks, they're very, very good at the image acquisition, the image segmentation, and all the feature extractions. You saw everything that Seungtaek showed you in his previous slides. But what we can do is take all of that data, relate it, export it to FCS Express for some simplified data analysis and report generation, and for communication and publication of your results. And that's kind of the end point that we really want to get to, is communicating and publishing that data. So I'm going to show you very quickly some of the things, how this kind of works in FCS Express. You'll notice that we're in FCS Express right now and that it kind of looks and feels like Microsoft PowerPoint or Microsoft Word. That's shamelessly by design. We want it to look just like Microsoft Office because everybody knows Microsoft Office, and if you can use Microsoft Office, chances are that you can use FCS Express. So what I want to show you in this layout, we have a plate-based assay here. This is an image that's just showing you what's going on in this particular nuclear translocation assay. I'm going to spare you the details because we have much more interesting scientific results going on today that are going to be presented a little bit later. But the idea is that we can view the multi-parametric data from your image is in a heat map display like this. It can be from one image per well to as many images per well that you can take. And what we can do is plot all of that data, so each individual cell in an image as a dot. We can do that in a scatter plot like this in FCS Express. We can choose different gates or different wells or pools of wells. So if we wanted to look at all of the positive controls in one scatter plot, and you notice that's what we're looking at here. And if we look at all the negative controls or the non-translocating cells, we can see that here. We have also access to all of the different multi-parametric data that has come out of this assay. So if we wanted to change to look at something like the mean intensity of the DNA versus integrated intensity, we can certainly do that just by clicking here. We have unlimited undo, so if I want to undo those changes, it's very quick and easy. But what this allows us to do is create gates, or what we call gates in flow cytometry, which people may be more familiar with them as thresholds in image cytometry, to say, let's look at all of the cells that fall outside of the translocated population. We'll call them gate 1. And we can call these cells, we can show this gate over here, and we can say that these cells are non-translocating. So based on that principle of setting up gates, what we can do is come over here and further interrogate this data. We have our non-translocating cells gate, and we have our translocating cells gate here in red. But we can also do things like insert images. So these are the raw images that came off of this assay. You can see that there's a cytoplasmic and a DNA channel. And we can also view the individual cutouts of individual cells, so all the way down to the single cell level. The way that this works is everything is interactive. All of the cells that fall within this blue gate are appearing as blue in these two different images. All the cells that appear as red are appearing as red in these two different images. And as we move one of these gates, you'll notice that these are dynamically changing. So we're able to see where these cells in the scatter plot come from in real time. So that can allow us to do things like find outliers. You can see here that this one little outlier cell actually appears here in the image. And we can even apply this gate just by dragging and dropping onto the image to see where that comes from. And again, we can bring that down to the single cell level. If I undo these changes real quickly, you'll notice that we also have the option here to work with different wells in the plate. Just by clicking, we can see how all this data is changing for us in real time. And that's a kind of brief summary of what we can do in FCS Express. Again, is setting up all these interactive gates and plots to get you to the end result of some very nice figures and presentations. All the data and all the analysis in FCS Express changes in response to these gates. So you can notice here how my regression curve is updating as we change this around. And if you have 100 plates, we can analyze them very quickly and easily, or if you have one plate, we can do that as well for a batch process. So with the basics of FCS Express kind of down, I wanted to hand the presentation over to Gianluca, Dr. Pegoraro, to show what he's been using FCS Express for and what he's been using the Columbus and all the PerkinElmer instruments for. So Gianluca, are you ready to take control here?
Yeah.
Great.
Can you see my screen?
Everything's coming through great. You can take it away. Thank you very much.
Thanks. Hi, I'm Gianluca Pegoraro, and I'm a research scientist at USAMRIID. Thanks, Sean, for the introduction. The main research interest at USAMRIID is the study of infectious diseases. And in particular, we are interested in quantifying host-pathogen interactions with high-content imaging. So today, I would like to give you a brief overview of how we integrated FCS Express 4 and image cytometry in one of our workflows, high-content workflows, for the characterization of pathogen-induced cell fusion. But before starting with the analysis, let me give you a brief introduction about our science. Okay.
certain intracellular pathogens have the capability of escaping the host cell phagocytic machinery and get access to the cell cytoplasm. Here, they can hijack architecture of cellular proteins like actin to promote intracellular motility. But what's most important for today's presentation is the fact that these pathogens can actively promote cell-to-cell fusion of infected cells. So the goal of our analysis is to identify the product of cell fusion, or which are usually referred to as multinucleated giant cells, or in short, MNGC. In terms of image acquisition, we use the Opera high-content imaging system. It's a confocal system. And we use the 20x objective, water immersion objective. And we use 4 channels, as you can see here. Here I'm showing non-infected cells or mock-infected cells. These are our negative control. And as you can see, we stain cells with the DNA stain first, shown here in blue, which emits in the UV channel. We also stain these cells with cell mass deep red to identify the edges of the cell body. Cell mass deep red emits in the red region of the spectrum, and it's indicated here in red. As I mentioned before, we were also interested in possible alterations of actin, so we stained these cells with colloidine conjugated to Alexa 568. So what you're seeing here in white is the cellular distribution of actin. And finally, we reserve the 4th channel, the green channel, for the immunofluorescence against one of the pathogen proteins. And so we can specifically detect the presence of the pathogen. Of course, these cells are not infected, so there's no pathogen. As you can see from the images, the picture change dramatically upon pathogen infection. And there are several morphological telltale signs that the pathogen has a strong influence on cells. The first one is that the nuclei of infected cells tend to congregate in these clusters, as indicated here by several white arrows. The second being that these cells that have nuclei clusters are also much larger than the non-infected cells. You can also appreciate that there's a dramatic rearrangement of the actin cytoskeleton in these infected cells. And we use this property to distinguish cells that are MNGCs from non-MNGCs. One important thing that I want to point out is that even though there is a lot of MNGCs in the affected sample, you can see that there's infected cells, but that are single. So our task is not going to be only identifying MNGCs from non-infected cells, our negative control, but also to distinguish them from other cells that are indeed infected but haven't fused yet. In terms of image analysis, we use PerkinElmer a cappella, and we designed a custom script to extract and quantify several morphological features that might be useful to distinguish MNGCs from other kinds of cells, both in the infected and uninfected cells. And of course, we look at the clustering of the nuclei, at the cluster size, the cytoplasm size, and the number of bacterial spots. And also, we look at actin texture and morphology, so the distribution of actin inside cells. I'll want to briefly walk you through the image analysis. As in most cases in high-content analysis, we started with the nuclear segmentation as primary object, and so we use the DNA stain channels to detect the nuclei. And as you can see, a cappella does a pretty good job in separating the nuclei even when they are close to each other. What you can notice, though, is that especially in these huge clusters of nuclei, the nuclei are really touching each other, are very few pixels away. This prompted us to to insert a nuclei clustering step. And so here we instructed the software to group objects that are either touching or very close to each other in one single object, as indicated here by the fact that the nuclei that were identified as separate objects in the previous step are now one single object. I also want to point out that this clustering step is pretty specific for the infected sample, but even in the not infected, in the negative control, we have few objects that have multiple nuclei, and we'll use FCS-H4, as we will show later on, to actually figure out these false positive images. The next step was using the cluster nuclei as objects, feeding objects, to expand the cytoplasm. And we used some deep red, the some deep red channel, to find the edges of the cells. And as you can immediately see in the lower panels, the infected sample, in the infected samples, the MNGCs tend to be, as expected, much larger than non-infected cells. But also you can see that there is other cells that are infected but not are not large. Then spot detection was relatively easy because, of course, the differences in terms of levels of infection between the non-infected and infected samples are pretty strong. We applied the spot detection in Capella to find the spots that are specifically associated or internalized in cells. Finally, we also extracted star morphology features. These are features that are calculated, as Seungtaek mentioned before, and that give an indication of the intracellular distributions of fluorescence. And we applied these star features to the actin channel. And in fact, you can see here there is dramatic differences between the infected and non-infected sample. And here in the lower panels, I'm just showing one of the many texture filtered images that were used for the texture analysis. So this is, as far as image analysis is concerned. In terms of outputs, we measured around 200 features with the analysis workflow. For today's presentation, I just wanted to focus on a few important ones. The area of the clusters, the number of nuclei per clusters, we can measure that in a Capella, the number of spots per cluster, and a few of the star morphology features that we measured in the actin channel. And also, I just want to point out that the Capella script automatically outputs all the necessary data, both at the well level and the object level, that is needed by FCS-H4 analysis. We then imported all the data that we produced in a Capella into FCS-H4. And the first thing we did was to get a high-level view of all our well-level data. As you can see here, I'm showing 3 heat maps of these example experiment that I'm showing. And we infected cells in a 96 well plate, and the first 2 columns were not infected, so these are our negative controls, whereas we had also 6 different strains of bacteria that we wanted to test. As Sean has explained before, here the green wells have low numbers of spots per cluster, and the red wells have high numbers of spots per cluster, or nuclei per clusters, or a large average cluster area, as expected. Another nice feature of FCS-H4 is that we can create well-based gates, and here we created well-based gates on a per column basis, so we could pull 6 independent replicates for the same sample. Here I'm just showing 2 of them, but we did the same for the 6 different strains and the 4 the 2 control columns. Finally, in terms of gating, we also applied 2 single well gates that are useful for image plot drawing, and here we gated one negative well here and one positive well here, as you see. So our task was to distinguish MNGCs from non-MNGCs based on the features extracted in a cappella. And the first thing we did was to create dot plots for both our negative control and for our positive control, this reference strain A, and to plot several different properties. We ended up choosing these two star morphology features because, as you can immediately see, the feature represented here are different between uninfected and infected. I also want to point out that here we are looking at single cell data or single cluster data. And so, in fact, here, every single dot represents one single cluster. Having said this, we define a quadrant. So we define the dot plot in quadrants, and you immediately see that in the infected sample, this is the quadrant in which there is an enrichment of clusters. We then created gates based on the upper right quadrant, and we decided to call this MNGC plus. And as you can immediately hear, the red objects are now part of the MNGC plus gate, so the upper right gate, and we have an increase from the negative control to the positive control. We also created other two gates. One is a complementary gate called MNGC minus, which represents everything that is not part of MNGC plus, so the remaining three quadrants. And then also another gate that we named clumps, which corresponds to the lower right quadrant. And you immediately see why we decided to call this gate clumps. These are feature plots of the two single well gates that we created. The upper one represents one negative well. The lower one represents one positive well. And what we immediately did was, in order to check whether our gates corresponded to the images that we were seeing, was to pseudo-color the clusters based on their on the fact that they were belonging to a specific gate. So you can immediately see here that these objects in the lower panel and the positive control colored in red are actually belonging to the MNGC plus, and they're large. They really look like the MNGC objects that we defined before, whereas the blue objects are defined as MNGC minus. And of course, in the negative control, there's way more MNGC minus objects when compared to MNGC, where compared to the positive control. Also, importantly, these dot plot gating strategy allowed us to filter out the false positive, the clumps that you can see here colored in green. So these are clumps that are formed even in the negative control due to the fact that these cells tend to be sticky, but we can definitely figure them out in subsequent steps of the analysis. We repeated the same for all the other strains, and you can immediately see here that the different strains create MNGCs at different levels. These four, the positive strains and these other three strains create MNGC at a high level, whereas these two strains here into form MNGCs at a lower level. But what we can also do with FCS4, now that we have two independent populations, is to actually look at other properties that we haven't looked at yet, and see if there are differences between the cells as we gated them. So as you can see here, this dot plot is gated on all infected cells. So here I'm looking at all the cells but for the negative controls. And in red, I plotted cells belonging to the MNGC gate, MNGC plus gate, whereas in blue we have the cells belonging to the MNGC minus gate. And it's immediately evident that MNGC plus cells are larger, as expected, and also have a larger number of nuclei. When we created a dot plot this time, and this time we used here a number of spots on the axis, we see similar behavior. So the cells that are part of the MNGC plus gate are not only larger, but also have a larger number of spots per cluster, which was also expected given the fact that they were much larger. What attracted our attention, though, was that even though these two populations, MNGC plus and MNGC minus, are fairly well separated in this region and at a low values of area, there is a region here in the middle where there's actually some overlap. So to get a better look at this region and the behavior of the two populations, we now created two new dot plots. And here I'm again plotting area versus number of spots, but this time I'm getting the two dot plots separately, getting the two dot plots on the two populations. So you can see here, these are only MNGC minus cells on the left-hand side dot plot, whereas we have MNGC plus cells only in this right-hand dot plot. As you can see here, there's very sharp, there's a very sharp drop of MNGC minus cells at this area value, so they're not larger than this value. And I haven't colored these in green, but these are actually cells that belong to the clumps gate, so we can ignore them. And at the same time, the MNGCs seem to be classified as MNGCs only from a certain value of area onward. But we have this population in the middle that it's sort of intermediate. So we decided to create a third gate that we named intermediate to describe the fact that these cells are overlapping and that have intermediate value of areas in number of spots per cell, and two other gates of early infected cells and mature infected cells. So we went back and checked whether these gating had really reflected what we were seeing in the images. We could see that indeed, and here I sort of colored the objects with the colors of the gate, early, mature, and intermediate. As I was saying, we can really see that cells belonging that are early infected tend to be small, mononucleated, and most of the time sort of more distant from other cells. We then have the cells that we colored in yellow that we labeled as intermediate, which already start to get larger and to fuse with other cells. And then we have the mature infected cells, which correspond to what we originally defined as the original MNGC population. So this is just to give you an idea of what we can do with STS Express 4 and a cappella generated data. So with this, I'll end it here. These are the people that participated to the project. I want to thank them. And now I'll hand it over to Krishna. Thank you very much for your attention.
Hi, Dr. Pegoraro. Thank you very much for the presentation. If there's any questions on this presentation, you can feel free to ask them in the questions box. We'll relate those over to him as we move forward. But what I'd like to do is move ahead here, and we're going to introduce Dr. Kota. I'm going to make you the presenter. Can you hear us?
I can hear you. Can
okay.
You hear me?
Very good. It looks like you're coming through, and you can take it away there.
Hi, everyone. I thank organizers Seungtaek and Sean for giving me this opportunity to present some of the research work that we are doing at the USAMRIID. The title of my talk is application of the image cytometry to quantify the host pathogen infections at the single cell level. So a brief introduction about the biology that's involved in this presentation. Since the proteins or the factors, the host factors that were involved in this work were not presented, we renamed the factors as host factor 1, 2, and 3, and the pathogen as pathogen X. Coming back to these factors, the host factors were recently shown to restrict several pathogens, and 3 of these factors, which we call as HF1, 2, and 3, show differences in their ability to restrict distinct pathogens. HF2 and 3 are 93% identical, whereas HF1 is truncated amino acids. And all the pathogens restricted by these proteins require access to acidified cellular compartments, and the current data suggests that HF may block release of these pathogens from the acidified cellular compartment. The question we are asking here is, what is the difference about HF1 from the other two HF2s, that is HF2 and 3, that alters its ability to restrict different pathogens? Briefly, the methods that we use, Vero cell lines, which are stably expressing the HF1, 2, and 3, or the vector alone were generated. All the cells were plated on 9612 format and imaged on Opera high-content imager. The infected cells were detected by immunofluorescence analysis of the pathogen-expressed proteins. And all the images are acquired with Opera high-content imager, either with a 10x air objective or 63x water objectives. Image analysis was performed with the Columbus image analysis system, and the data analysis was done using the FCS Express. So in this figure, you see the Vero cell lines expressing either the vector alone or the host factors were infected with the pathogen. On the top left panel, you see the cells expressing vector alone are infected nicely with the pathogen. So does the cells expressing HF1. But the cells which are expressing either HF2 or 3 are not infected with the pathogen. This shows that the cells which are expressing HF2 or 3 are restricting the pathogen infectivity. And again, in this slide, we want to see the localization of HF proteins inside the cells. These are below stable cell lines expressing HF protein, HF2 protein. And then the cells were transfected with the either HF1 or HF3 or HF2 plasmids, which are MIG-PAG. And as you see here, the cells which are transfected with HF1, the cell lines are not infected with, the cell line forms huge vesicles, and also they are not colocalized with HF2, whereas the HF2 and 3 are colocalized. So the goal of this project is analyze how HF expression levels alters infections at single cell level. The second thing is we want to quantify the morphological changes. Like as I shown in the previous slides, there are large vesicles formed when the cells are expressing HF1 protein. And we want to see if there is any change in the number and the size of these vesicles. This is a well view of the cells, a well view of the cells infected with the pathogen. The green spots you see are the cells infected with the pathogen, and this image is obtained from the FCS export, which have access to the image analyzed and stored in the Columbus. One of the nice features of these FCS exports is we can also view the cells at the single cell level, as mentioned by Gianluca earlier in his presentation, and we can also see many of the features. In this slide, I'm just showing few of the features, but there are many features that we can see at the single cell level. The other important thing is we can see the cell morphology or the mass of the cells, and here I'm just showing the cells which are infected with the virus. So the question we are asking is, we want to see what is the expression pattern in the cells expressing these host factors. And as you see, in the mock infected cells which are expressing host, the vector alone, there is a background, little bit of background, and we use these gating parameters in our future experiment to calculate the percentage of cells that are infected. In this slide, you see the cells which are expressing vector alone. On the left, on the top left quadrant, you see there are around 53% of the cells are infected. And on the x-axis is the expression level of the pathogen protein, and the y-axis is the expression level of pathogen protein, while on the x-axis is the mean intensity of the host factors. In this top right corner, you see the cells which are expressing HF1. There is no restriction on infectivity of the pathogen. We see around 63% of the cells are infected in the cells expressing with the HF1. When the cells are expressing HF2 or 3, the infectivity was reduced to 30% and 35% respectively, whereas in all these cell lines, like the HF1, HF2, and HF3, the 99% of the cells are expressing these proteins. So to summarize the previous data, the cells which are expressing vector or HF1, we have around 50% of the cells getting infected, and the cells which are expressing HF2 or 3, the infection rate was reduced to 30%. So in the previous slides, we have seen that the cells which are expressing the HF1 or vector alone, the mean intensity is lower compared to the cells which are expressing HF2 or 3. So this shows that there are multiple populations of cells in various cells expressing the various protein factors. So in order to measure those populations, we created a gate based on the cells expressing vector alone. And as you see, the cells and gave a cutoff of around 600 mean intensity. And here, the cells which are expressing vector alone has high intensity pathogens at the rate of like 2%. Similarly, when the cells are expressing HF1, the percentage of cells which have high intensity for the pathogen are around 2%. But the cells which are expressing HF2, they have 13% of the cells, 13% of the infected cells have high infectivity or high protein level from the pathogen. Similarly, cells which are expressing HF3 has 8% of the population has high pathogen intensity. So if we normalize this data with the percentage of infection, the cells which are expressing vector alone or HF1, there is only less than 5% of the cells are expressing high pathogen protein, while the cells which are infected with HF2 or HF3, the percentage of infection, they are over 30% of cells of high protein expression from the pathogen. What does this mean? This means that the cells that are infected or expressing the HF1 and vector alone, all the mean intensity of the pathogen is low compared to the cells which are expressing HF2 and HF3. That means the protein is able to be formed in the cells which are expressing HF2 and HF3, but somehow this protein is not utilized in the pathogen formation or the pathogen egress. So the assembly of the protein for the pathogen formation is inhibited. That's the conclusion we can draw from this analysis, which was only possible by gating parameters provided by the HCF expert. So the conclusion we can draw is HF2 and HF3, but not HF1, restricts the pathogenic infection. And the second is, among the infected cells, HF2 or HF3, 30 to 35% of the cells show higher expression levels of pathogen protein. And as I've shown in my earlier slides, the cells which are expressing these proteins, the HF1 especially, has a large vesicle size, apart from other acidified compartments. So in this slide, which is a live cell imaging done using OPERA, the acidified compartments are labeled with lysotracker red. So, or the lyso tracker green, which is shown as the in the bottom panel here. The nuclear are stained with the host dye. The cells which are expressing vector, there are few acidified compartments as shown red spots here. The cells which are expressing H1, the vesicle size increase enormously compared to those cells which are expressing HF2 or 3. Not only that, all the cells which are expressing the host factors have more acidified compartments compared to the vector alone. So the question we are asking in this analysis is, can we quantify the morphological changes in the acidified compartments induced by HFs? So the method is we acquired images of the V-row stable cell line expressing either of these proteins or vector alone, and they were stained with lyso tracker red or lyso tracker green dyes, and the image analysis was done by Columbus system and the data analysis by SPS Express. So the morphological features that we are interested are these acidified compartments, which are shown as red dots, and also the vesicles, which are shown by the green arrows. So there are two types of image analysis we performed using the Columbus. The first one is the spot analysis, which characterizes the number of acidified compartments. And the second thing is the texture analysis, which estimates the area of the vesicles. So characterizing the number of spots was easy using the spot analysis, but after trying various features, the texture analysis was the one optimum for describing the areas of the vesicles. Especially the whole feature of SER texture analysis, which Seungtaek talked earlier in his presentation, was more important in measuring the area of the vesicle. So this is an image analysis pipeline that was used to measure the texture analysis in Columbus. And this is a well view of the cells which are expressing large vesicles. And this is the distribution of the cells, of all the cells which are expressing either the vector alone or the HF1, 2, or 3 host factors. As you see from this graph, most of the cells which are expressing HF1 has larger vesicle size, which is shown on the y-axis, and the number of spots are more in all the cells expressing the HF factors compared to the vectors. I know this is a busy slide, so I'll explain in detail in these slides. So the cells which are expressing vector alone, there are very few spots, but not only that, the size of the vesicle is small compared to the cells which are expressing HF1, where the number of spots increase, but not only that, the size of the vesicles also increase. But the cells which are expressing HF2, the spots which corresponds to the acidified compartment increase compared to the vector alone, but the vesicle size is smaller compared to the H1. But HF3, the number of spots increase, but so does the vesicle size, but the vesicle size is smaller compared to the HF1. And this is what the FCX provided image looks like. The cells which are expressing vector alone, the spots number is small compared to the cells which are expressing HF1, where the vesicle size is also increased, so does the number of spots. But the HF3 is the one which has a more number of spots in, compared to the other host factors. This is a heat map. Again, the cells which are expressing HF1, their vesicle size is larger compared to the cells which are expressing HF2 or HF3 or the vector alone. So the conclusion that we can draw from this part is we could quantify the changes in the morphological features of vesicular compartments using the FCS analysis. We can also we can also mention that the host factors increase the essified vesicular compartment in the cell, and the difference among the host factor membranes in vesicle expansion was also noted. The increase in the vesicle size was as follows. HF1 has more vesicles compared to HF3 and HF2, whereas the number of vesicles are more in HF3 compared to HF1 and HF2. So in summary, the integration of OPERA's robust and high-quality image acquisition and Columbus modular imaging analysis and FCS image cytometry grading tools helped us in identifying the subpopulation of cells expressing differential amount of host factors. And the second thing is the Columbus texture analysis tools in combination with FCS image cytometry help us to visualize and quantify the morphological changes in the subcellular compartments, which are the acidified compartments in the cell. With that, I'll end this presentation. I would like to acknowledge Dr. Rajim Utsani, whose research work I presented here. I'd like to thank Rekha and Sina Bavari and Seungtaek from PerkinElmer, who was a great help for us in doing many of our image analysis and the setup at the USAMRIID, and also Sean Burke for helping us with the questions with FCS experts. Thank you.
Great, thank you, Dr. Kota. We actually have a question for you here that maybe you could answer. If anybody has any questions for any of the presenters today, feel free to type them in. But we had a question from Marjo. How long do you incubate the cells with pathogens before fixing and staining them? Hi, Dr. Kota, are you there? Can you hear us?
Around 24 hours. The cells are infected with the pathogen for 24 hours before fixing.
Great, thank you. And again, if there's any questions, please feel free to enter your questions in the questions and answers box there. We're going to just summarize a few things that you saw today from the De Novo and from the PerkinElmer side. I wanted to give you a quick wrap-up of what you saw with FCS Express. Essentially, what we're going to do is when that data comes off of Columbus, we're going to leverage all of the most powerful and proven tools for flow cytometry analysis with a lot of new imaging features to help improve that cytometry analysis. You're going to be able to move from population-based levels down to the single-cell analysis, and you're going to be able to interact with all the data between plots and images and cell galleries. We have a very intuitive Microsoft PowerPoint-like feel for the software, so it's going to be very easy to use and understand for any level of user. And we also have many resources for customers, like tutorials, sample data, online forums, a really great customer support system, and webinars like the one you're attending today. They're going to help you get up and running with FCS Express in a very quick amount of time. We also offer a free 30-day trial, so if you'd like to visit us at denovosoftware.com, you can go there and get the trial of the software. You can get some of these scripts that are provided by PerkinElmer and for building blocks or Columbus to export that data to help get you started there. You can also write us at support@denovosoftware if you have any questions. We will get back to you very quickly with any of the questions that you may have about today's webinar or any questions about FCS Express or image cytometry in general. And I also wanted to mention that on the De Novo software website, you'll notice that we have our webinars, our upcoming and recorded webinars section. After the webinar today, you'll be able to come to the recorded webinar section and view this webinar again if you'd like. And our download, of course, for the free demo is up here. And I also wanted to send this back to Seungtaek, if he'd like to give us a little wrap-up of everything that we saw from the PerkinElmer side of things. Seungtaek, if you'd like to take that away.
Yeah, sure. Thank you, Sean. Yeah, thank you, Dr. Pegoraro and Dr. Kota. I think your presentations clearly shows the resurgence or renaissance of phenotypic screening and drug discovery that we're seeing in pharma and academia these days. And we feel, PerkinElmer feels that our strategy is very well aligned with that approach, and our imaging portfolio, especially our Columbus system, is able to provide the sophisticated morphological, phenotypic, texture analysis that's very easy to access and use. Just want to thank everyone for participating. If you have any questions in regards to the Columbus or any of our high content screening technologies, please feel free to contact us. Appreciate your time.
Great. And again, thank you, Dr. Pegoraro and Dr. Kota, for joining us today for this webinar. And thank you again, Seungtaek, as well. We'd also like to thank everybody who has attended the webinar. I hope everybody was able to learn a little bit about the technology and about the science that's going on with all of these products and interactions. So again, if you have any questions after the webinar, please feel free to contact either the team at Novo Software or the team at PerkinElmer, and we can certainly get back to you with any questions that you have. So thanks everybody, and have a great day. Take care. Bye-bye.